as a human race, we have made some incredible strides in the fields of science and technology. We have discovered some really incredible things, such as induced pluripotent stem cells. My name is Jenna Hooten, and over the past few months, I've done some research on this really interesting topic in science. And using Amarillo College library databases, I have gathered some current research on this field of study. Basically, what induced pluripotent stem cells are is a body cell that has been chemically or other methods changed into a stem cell. So these body cells will look and behave like a stem cell does. They are pluripotent, meaning that they have the ability to turn into any type of body cell. For simplicity's sake, I'm going to call these iPSCs for the remainder of this presentation. The way that these body cells are turned into these iPSCs, you can induce them through chemical mixtures or through what are called transcription factors, which are simply proteins that change gene activity in the cell. So they will turn on and off genes based on what the cell needs its function to be. So these transcription factors named OCT4, SOX2, KLF4, and c-MYC are just they are inserted into the genome of a retrovirus, and this retrovirus will infect the cell and insert its own DNA into the DNA of the cell. And when the cell replicates, it will take that retroviral DNA and implement it into its own cell's genome, thereby changing the cell from a body cell into a stem cell. As you can see, the transcription factors are added to the adult cells, and then from the iPSC cell state, they can turn into any type of body cell. These cells are really useful currently in three major areas. Right now, researchers are using them for disease modeling. So they will take cells that have a particular disease, they will turn them into iPSCs, and then from their stem cell state, they will watch the cell differentiate, and so they can get a better insight into how these diseases progress at a cellular level. Another use for these iPSCs are through drug development and testing. So researchers can find the most effective and safe drugs to treat these kinds of diseases, again at the cellular level, without having to go through a giant experimental process. They can just look at the cells, which is the basic mechanism for bodies to work. And another way that these cells are useful is in regenerative medicine. And so one of the studies that I looked at actually proved that these iPSCs significantly decrease the time it took for wounds to heal. And so these iPSCs can be used for wound healing, spinal cord injuries, diabetes, and several other really large, really dangerous diseases. So these iPSCs can be used to actually treat those diseases. In addition to the more tangible uses of these iPSCs, there's a lot of intangible uses for them, such as these iPSCs overcome the ethical concerns that are associated with embryonic stem cells. So when embryonic stem cells were discovered, there was a lot of political fighting about this topic because in order to harvest these stem cells, you have to disrupt the life of an embryo. And so there was a lot of political fighting, and progress could not go very quickly because of all of the stigma that there was around this type of science. But iPSCs completely overcome these concerns because they come from normal body cells, so you can take them from skin cells. And because you can take them from any cell, there's an endless source of these cells, unlike embryonic stem cells, which you can only harvest one little set at a time. And again, because these cells come from the patient, they're patient-specific. And so there is little to no potential danger of a person's body rejecting a stem cell, an iPSC treatment. So if you take a body cell and, for example, turn it into a kidney, the recipient of this kidney's body is not going to reject it because it's made out of its own cells. Some obstacles that researchers are currently facing with this field of science are some safety concerns in the differentiation process. Because retroviruses actually mess with the DNA of a cell. There's a lot of genetic manipulation going on, which is a really complicated process, and it leaves open a lot of doors for things to go wrong, a lot of mutations to occur, and there's just a lot of space for things to go wrong right now. A lot of these cells are turning into tumors, which are not good. People don't like tumors, rightfully so. Another concern is inequivalency. Because some of these cells don't turn into the body cells that researchers are inducing them to become, there's the concern that these iPSCs are not equivalent to embryonic stem cells, which have been proven to fully differentiate. So there's the concern of the potential differentiation abilities of these iPSC cells. Another concern is the low efficiency researchers are finding in the differentiation of these cells. And so getting these cells to fully differentiate without causing tumors at a low cost is a pretty big issue right now that researchers are trying to overcome. And so the potential for these tumors to form can cause mutations, which can cause several abnormalities at the molecular basis of life. That comes from a study by Rajkumar Mongar. And if mutations are caused at the molecular basis of life, if cells can't work on a molecular level, the body can't work as a whole. And so that's a pretty big concern that researchers are trying to overcome right now. Currently, research is focusing on really understanding the methods of cell differentiation. And so the general thought is, if we can understand why these cells are differentiating and what is going on, then we can determine how to replicate this process chemically or unnaturally. And just really understanding the efficiency of this method and improving our methods of creating these iPSCs. Future research should really focus on perfecting methods that are alternative to using retroviruses, specifically such as chemical induction of these cells. Using chemicals to create iPSCs is going to be much safer because you're not messing with the genes of a cell. It's more time efficient, it's easier to control, simpler to use, and once you find a viable chemical mixture, you can produce that chemical mixture on a large scale for a really low cost, which is more efficient as a whole, and it's more safe as a whole. For as young as this science is, it's only about 9 years old, we have come so far, and we've discovered a lot of amazing things about our body and how it works. And I think that within the next 5 to 10 years, researchers are really going to be able to make even more incredible strides. And I think that these iPSCs will be used quite often for a lot of different uses.